Good day everyone, welcome to the course on biological data analysis and visualization with R. We have been working on a data set and we are performing WGCNA analysis. So, we have done several steps already. So, we have taken the raw data, we have normalized, we have then done the gene filtering, and then in the last class we did the soft threshold determination. So, this is something that actually determines what is the power value that will take the network to a scale free topology, and then we talked about the network generation part, right. So, this is something we had completed in the last class. In this class we will be continuing that analysis and will end that analysis. Okay, so this is what we are going to do in this class, right. So, we will first identify module of interest. So, we have already determined the different modules that are represented by different colors, right. This we have seen this and we have stored that data, and now in this class we will actually from that analysis we will identify modules of interest. So, when I say modules of interest, that means these modules show different pattern or different behavior across different samples, right. So, we are working with cancer versus normal sample. So, these modules show different pattern in cancer versus normal. Okay. So, these are modules that are likely to be important for cancer. Okay. So, once we do that, we identify some of the modules, we can look into the expression profile of genes, and this is something that is probably very important. And then I will very briefly talk about network visualization, but will not do this in this class because of the time constraint. It will take a lot of time actually to load this into a visualization tool and to analyze the network or visualize the network. Okay. So, once we visualize the network, you can see that some of these very interesting properties will emerge. Nevertheless, I will just mention how you can do this visualization, and this is something you can definitely try. Okay. So, this is a flowchart that we are familiar with. So, depicting the WGCNA workflow. We have talked about the first 5 steps already in the last few classes. We started with data loading, normalization, gene filtering. In the last class we talked about soft threshold determination. We have seen we can test different thresholds using the peak soft threshold function of WGCNA package, and it suggests some threshold, but it is not necessary that we will have to peak that threshold. We can then do some plotting and visualization, and we can peak the threshold that we think takes us closest towards the soft free topology, but at the same time we want the minimum threshold where we reach that soft the scale free topology, right. So, we have seen this in the last class. We did some visualization, etc. And then once we choose a threshold, we then use that threshold to do the network generation. Again, this is something we had done in the last class. We have generated the network, and there were I believe 18 modules in total that were identified, and these 18 modules are represented by 18 different colors, right. That is how we stored the data again. So, this is something that you can use and we can visualize the results. What we will do in the next class, we can of course in this class is of course we can store the network data, and then we can identify modules of interest and then look into the gene expression profiles in the modules. So, what are the modules of interest? As I said, modules that differ in their pattern among sample types or different treatments. Okay. So, this is something that is very important because here we are looking at into cancer versus normal samples, and there are modules that will show different behavior. Okay. And this is something that we would like to know what are these modules and what are the genes that are there in those modules, okay, because these genes are likely to be important for cancer development, progression, etc. Okay. We can store the network data in something called topological overlap matrix. Okay. So, this is something you have seen already, right, the TOM files. When you generate the network using WGCNA package, we generate these TOM files or topological overlap matrix, and this is something we can store. Okay. So, what do we have in topological overlap matrix? It kind of describes the connectedness of genes and overlap in the neighborhood in the network, right. So, what is the overlap between two genes in the neighborhood, right. So, one gene is connected to multiple other genes, and if you consider a second gene, that is also connected to other genes. When I say connected, they are kind of showing expression correlation, right. So, when you say overlap, right, so you want to see, right, whether there is any overlap between gene 1 and gene 2 in terms of correlation with other genes in the network. So, this is something that is stored in the topological overlap matrix, right. So, this kind of gives us an idea how genes are connected in terms of their expression pattern, etc. Again, this is in broader perspective, right. This kind of tells us, okay, maybe these genes are co-regulated, right. They are kind of have the same regulator, transcription factor, etc. And those could be really interesting for understanding of the biological process. Okay, so with this background, let us now move into the hands-on part again, right, and we will complete this analysis in this class. Okay, so let us move on to our console and let us do the hands-on again. Okay, so the network that we generated in the last class is stored in this network file. It is called network, right. And as I showed you, right, there are MEs, right, 18. So, this is something we have seen already, and then we have, as I told you also, we have 2 dendrograms. So, dendrograms are simply connections, right, the connections between the, or how close these genes are, or the modules are, right, in terms of their patterns, right. So, that is something that is stored in these dendrograms, and we have visualized some of these dendrograms in the last class. Tomfalls, as I mentioned, this is what we have information about them, right, the topological overlap, and the block genes. Again, we have 2 such sets, right, and this is something we are going to visualize today, okay, and identify the modules of interest. So, for doing that, what we will do is something called module eigengenes. Okay, this is something that we are going to use. This is a function that we have in WGCNA package. Okay, so what this module eigengenes will do is, okay, so let us run this first and then we will see. Okay. Okay, so what I will do first, let us run this, then I can describe what it. Okay. Eigengenes. Okay. Okay. So what we have is kind of like the probably the most important genes, right, that are kind of showing these expression patterns, right. So this is what we stored as the eigengenes. Okay, it's kind of like the most important genes of the hub genes. Okay. Okay. So, for each color, which means each module here, so ME black is one module, ME blue is one module, ME brown, etc., and these are the samples, right. So, we have this expression of probably the most important gene or the hub gene, right, which we call as eigengene in this analysis, right, and this is something is stored in this eigengenes function, okay, eigengenes value. Okay, so we have seen these eigengenes. Now, what we can do is we can order these modules based on expression or the importance of these eigengenes. Okay, so this is something that's what is happening here, right. The order these MEs, and this is a function from WGCNA package, and we are taking these eigengenes here and we are sorting them. So, which is kind of like telling, okay, this function is likely to be most important and this function is likely to be or this module is likely to be most important, etc., that kind of idea. Okay. So, this is the order modules now we create. So, what we are getting is these names of eigengenes, right, and here. So, these are the module names, right. What we are doing here is we are simply taking these names of these eigengenes and replacing this ME with blank. Okay. So, we do not want this ME blue or ME black, etc. We just say blue, black, etc. So, this is kind of simply an change in the names of the columns. Okay. Okay, so it's kind of very similar idea again, right? We are changing the names of the columns here. Okay. So now we have identified these eigengenes and we have ordered the modules. What we are going to do is now identify the most interesting ones. Okay. So we can do this simply from these eigengenes, right, and see which modules are the most interesting ones to us. Okay. Okay. So you will use a heat map, right, to actually show this, see this pattern of these or the values of these modules across different sample types, right. Okay. And for generating this heat map, right, we will use this gplots package, and we have this as matrix eigengenes, and then this is where the heat map function is, which is from this gplots package is the heat map dot 2. Okay. And this will take only a matrix, so that is why we are doing this matrix function, right. And then of course we have a lot of other parameters or arguments, right. So this is something I want to show you what this means here. So heat map 2. Okay, so let's see this here. Okay, maybe not. Gplots package. Okay, it's probably anyway. So perhaps we can find the gplots package here. Okay, and yeah, gplots we have. We have heat map 2 function. Okay, and here you can see this heat map function. There are a lot of things that have been discussed. For example, what does this row V column V means, right? Whether we should have clustering along the rows or the columns, etc. Whether there should be dendrogram shown, right? That relationship between the samples, and whether what kind of color it should be using, right? Whether there should be trace, etc. So this is something you can explore, but for now I'll just use these arguments, right. This gives us the best viewing results. So you will see I am saying row V is false. We don't want any clustering, we don't want any dendrogram, we do not want any trace, and then the color that we want is the red blue 50 scale. Okay, so this is what we want. Okay, so red blue color will give you this kind of plot. Okay, so you can use other different color formats as well. There is red green format, etc. So we are using this red blue. Okay, so this kind of tells us the different values of these modules. Okay, so just to interpret this plot heat map, what we have along the rows are the sample names, right. So here you have the tumor versus normal, and here you have the modules that we have identified, right, from the network, and there should be 18 of them, right, and these are the patterns, right. So the values of these modules are given here in the color key on top in the histogram here. What we want to see here is, okay, we now want to identify the module of interest. Okay, what is a module of interest is so a module of interest is a module that show different value or different pattern between cancer and normal samples. Okay, so as you can see probably here, this set of modules here, they are mostly red in color, whereas in the normal samples, right. So this lower part is, you can see the N here. So this means in normal samples they show red pattern, whereas in the tumor sample they show blue pattern, right, blue values, right. So positive values here and negative values, mostly negative values in the normal samples. So there is difference, right. And if you look at this set of modules, you see they are showing red color in the tumor samples, negative values in the tumor samples, and the blue values in the normal samples. Okay. So, there is again this difference in pattern, right. This pattern is kind of, you can see. Okay. So, what you can do is we can pick two modules here that looks most interesting, right. When I say most interesting, they are all throughout normal they are red, and in tumor they are blue, and we pick another module which is here, right. It is blue throughout the normal samples and red throughout the tumor samples. Okay. So, this is something we want to see. So, let us pick two modules, anything we can pick, right. So, maybe we can pick green here, right. This green module here is blue, mostly blue in the normal sample and mostly red in the tumor samples, and similarly here we have, let us say, the brown one. Okay. So, which is actually red in the normal and the blue in the most of the tumor samples except this one. Okay. So, we can pick anything we like, but to me it seems like green and brown. Okay. So, or brown or turquoise. So, maybe I can change this to, let us say, brown and green, okay, as we discussed. So, anything you looks interesting to you can take two. So, it does not have to be two, it can do more, but for the sake of time we are picking only two modules here. Okay. So, we have picked two modules now, right. These are the modules, interesting modules to us, right, from the heat map. Okay. And this is what we have. Okay. So, what we are going to do next, okay, so is we are going to look at genes that are in these modules. Okay. This is what is interesting to us, right, and what is the expression profile of genes in these modules. Okay. So, before we do that, let us save this. Sorry, file equals to the usual function, right, and okay, there is a spelling issue. Okay. All right. So, we have saved this. Now, let us look into these interesting modules, okay, that we have chosen, and let us look at the gene expression profile inside these modules. Okay. So, we have now identified the modules and that look interesting. We have kind of arranged them again in order, right, which we have done in the earlier part, right. We have ordered these modules, right, based on this order ME function, right. We are reordering this, we have identified the eigengenes, etc. Okay. So, now we have, once we have done this, now we are going to look at the expression pattern. Okay. So, this is what we are going to do. Okay. So, what we are doing is we are now subsetting creating a subset of modules, right. So, again this function we can use for more than two, right, and this is, I hope you remember, right, this is what we are getting from DPL via diverse, right, package. We can do this. So, we have the modules list. From that we are doing a subset for which the colors here, right, colors means the module color is present in interesting modules, right. So, these interesting modules are here, brown and green that are mentioned, it could be even more than two. So, all those modules will be picked and stored in this subset. Okay. So, once we run this, let us, okay. So, again you have all these genes that belong to these two modules, right. You can see this either green or brown color. Okay. So, we have these now chosen this subset of modules. Okay. And then what we see here, these are the row names modules, that is the gene names. Okay. So, okay. So, that is the gene names of the row names we have put there. What we are going to do is now we are looking into this subset gene expression, right. As we said, we want to now look into the expression pattern of these genes that are part of these modules, right. So, what we are doing is from subset mod that we have created, the subset of modules that we have created, again we have some functions that are available, right. So, these functions are $genes and $colors, right. So, $genes will give us gene names and colors will give us the module membership, right, whether it is green or brown in this case. So, what we are doing is from normalized data, right, this normalized selected data, we are taking expression values of those genes that are part of this subset module. Okay. This is what this means, right. So, this part means, right, we are taking those rows that belong to these subset modules, right, these genes, and then we are storing these expression values in the subset gene expression variable, this variable. Okay. So, what we have is okay. So, right. So, this is what we have done. Okay. We see that these genes we have taken and we have now the expression values in all samples, across all samples. Okay. These are the normalized values, right, that we normalize variance stabilized values that we generated, and only for those genes that belong to brown or green module here, right. So, you can see, for example, we have ISG15, we have this GABRD, this gene, because this also belongs to brown module. Okay. So, only those genes have been picked and their expression levels across all samples in our data set. Okay. All right. So, now we have taken this gene. Now, what we are going to do is we are kind of transform this data a little bit so that we can plot, okay, using ggplot. Okay. And that is what we are doing, right. You have seen these functions when we are talking about data organization, right. We are taking this data frame and then we are mutating these genes, right. So, row names, and then we are doing pivot longer function. Okay. So, you will see this what actually each of these functions will do, right. So, you can actually try each of these functions and see what actually happens. Okay. This is something that you can try out. So, again we have discussed, right, all of these functions when you are talking about data organization, right. So, the mutate function, the pivot longer function, all these functions we have talked about when you are talking about data organization with DPLYR package, right. Now, what happened is simply we have transform the data, right, or reorganize the data, right, in a table format now, and you can see that the whole thing has changed, but what we have now is the gene name, right, this is character, the name of the sample here, the value, right, value of that gene in that sample, the expression value, and the module that it belongs to, right, that is what we are getting here, right. So, it is only the data has been kind of transformed, right, in a specific format. Okay. So, that is what we have now. Okay. Again, we can generate this in different ways to get to this format. Okay. So, we use this format now to use the ggplot function. Okay. So, again we have used ggplot2. So, this is the data that we will use, then we have the x equals to name, y equals to value, right. From this data format you can see x equals to name, right, that is the sample name, y is the value, right, the expression value, and then groups are the genes, right. So, based on the gene value. So, you have to group the person. Then you have the geome plus geome underscore line, right, plus this is the geomes function, part of the geome function, right, and then we have the plus theme minimal, right, again this is the theme of the plot. You can have this axis text, what should be the angle of this text, right, this is put at 90 degree because these names are really long, and then we have the grid, etc., the what should be the labels. So, this is what we are it is what we are doing, right. So, the labels should be x should be the samples, right, and y is the normalized expression, right, that is what we are looking at here. Okay. So, let us look at this, right. So, maybe we can try this perhaps. Okay. So, what it is suggesting, okay, so because we need to complete this with all of this, let us try it. Okay, so it is actually probably giving us some error because of some problem with library. Okay, so perhaps we have to restart the whole thing. So, that is why it is giving this error. Anyway, so this is how you will actually get a plot, which we are not getting right now because it is saying something wrong with the installed library here, and that is something causing a problem. Okay, so which means we have to restart the whole console and set this up again through all the analysis. So, which again will take a lot of time. So, I am not going to do that, but this is something you should generate a plot, right, which will show you the expression value of these genes with different samples, right. So, and in different samples. Okay, so this is something we will not be able to do here. The other thing that we will be able to do is the store the network. Okay. So, the store network part is using this the last part, right, which we will use to store the network, right, and these are the interesting genes, right. So, we have stored the interesting modules here in this value here, okay, and then what we are doing is we are doing a subsetting again from these modules and identifying the list of interesting genes. Okay, maybe you have to close this first and then we'll okay. Okay, okay, yeah. So, this is fine now, right. So, we have identified these interesting genes, okay, and what we want to do now is we want expression values of these interesting genes again from the normalized data, the norm data. So, what we are doing is we are reading the expression values from the row values. Okay. So, we can look into all of this, right. So, these are the names of the genes, and this is again we are using this subset function, right, on the modules, and we are identifying these genes. We can also look into the expression values that we have identified, right, for each gene into different samples. Okay. Now, what we have to do is we have to calculate these topological overlap matrix, okay, and this is the function again from the R package WGCNA, Tom similarity from expression, okay, and this will generate these Tom values. Okay. So, this is what we are getting from expression values, it will generate this topological overlap matrix, okay, and we are taking this expression value, we are doing a transpose, we are using the power here, the chosen power, which is 22 here, and then we are using this Tom similarity from expression matrix, okay, for, this function. Okay. Again okay. So, because we have I do not know how many genes we have, right, let us see, yeah, we have about 729 genes, right. So, then we so we are getting this Tom the Tom value, right, that this is calculated now. Okay. So, once we have this Tom, right, we can see this how this looks like maybe, right. Okay. So, this is the matrix. Okay. Perhaps what we can do is we are actually what we are getting here is the correlation value mostly, right, and this is what we have row names Tom, right. These are the row names should be the gene names as well as the column names, right. So, now once we have replaced this, let us see. Okay. So, what you see is the row names are the gene names as well as the column names. These are also gene names, but only for genes that are of interest to us, right, that are part of the interesting module, right, which we have chosen in this analysis to be green and brown only. Okay. From that, now what we will do is again we are transforming the data, as you can see here we have the data frame, then we have muted gene 1, right, pivot longer, again we have used this function. So, what we are doing is we are simply converting this into a list of edges, right. This is a network. So, which means it should be only edges, right, connection between genes, right, and that is what we are converting these to. Okay. And these connections we are creating with again this DPLR different functions, rename, etc. As you can see, we are doing subsetting where we are not choosing when gene 1 is equal to gene 2, we do not choose that as an edge, right, we said self edge, we are not choosing that here, and we are also mutating this module 1, module 2 colors. Okay. So, let us look at this, sorry, list edges, okay, and here you see this. Okay, so here is the gene 1, gene 2, that is the correlation value, and the module 1, module 2 membership, right, which are whether gene 1 is part of module 1 and whether gene 2 is part of module 2, right. So gene 1 here is part of brown module, gene 2 is also part of brown module, but you also have brown module, green module connections. So all these things are stored in this file, okay, in this list edges. Now, why you store these list edges is we can now write this list edges variable as a file, right. We can export this as write.table command, and once we write this, now we can then use these for processing with other tools like Cytoscape. Okay. So, we can use network visualization tools like Cytoscape to actually visualize the network. We are not going to do that because this is going to take a lot of time to load this file and visualize the network, but you can try out using different tools. One example is Cytoscape, but there are other tools that you can also try. Okay, so with that we have completed the analysis, right. We have gone through all the steps that I mentioned, right. We have stored the network data, we have identified modules of interest, and we have also identified gene expression profiles in model, but of course we could not visualize because of an error in the R library. That is something probably will go away if we try again, but of course we are out of time. So with that we have identified modules of interest starting from gene expression data. We have identified modules of interest. We have also stored the some of the expression profiles of these associated genes. We could separate out the expression profiles, and then we have seen some of the visualizations, right. Of course, we have not seen the network visualization, but we have seen the modules, right, and we have identified some of the modules of interest. But something that you can do is the network visualization and network analysis, which will be the further steps, because this will give us an idea about the network properties, how the centrality of the network, centrality measures, etc., are changing between normal and cancer sample. Thank you.